Thank you very much for being with us.
Thank you very much for the introduction. It's a pleasure for being here. Thank you very much. I think this was the most straightforward invitation I've ever experienced. My name is M Meier. I'm a member of the interdisciplinary spinal research at the Balgrist University Hospital. So we are a part of the chiropractic medicine department, but we are very interdisciplinary. So we are human scientists, neuroscientists like me, movement scientists, biologists, and also we do also genetic investigations. You can see or have a look here at the homepage. And today I would like to talk about a psychosocial issue, fear of movement, fear of pain, and why are these fears important for clinicians and researchers, and especially for patients also. So this is the outline. I've heard minutes ago that you already had a sophisticated introduction in the fear avoidance model, so I will go over that shortly. Then I would like to talk about the assessment of fear of movement and fear avoidance beliefs, and the different questionnaires which exist. And then go over to supraspinal correlates of fear of movement. Actually, I'm doing brain research, and I'm interested if we can dissect the fear, this sort of fear, in the brain. And if we see brain correlates of fear of movement, of this phenomenon. And finally, I will come to a conclusion. So, some of you know this, or this is the original model of flying. I will go over it shortly. So this is the fear avoidance model. So first there is an injury, and if you go to the right, so this is the normal case, you experience no fear. There will be a confrontation, and finally a recovery. But dependent on the affective state of maybe of a vulnerable person, the pain experience can lead to negative affectivity and to catastrophization. And this leads to pain-related fear, which finally ends in avoidance behavior. And especially in chronic low back pain, I will talk today about, as an example, chronic low back pain, because we mainly investigated those patients. This cycle will end in a vicious cycle in the end, and will lead to pain amplification because of the disuse and the disability. And finally, the pain experience will be not anymore related to the initial injury. It will become a unique entity for itself. So that's the fear avoidance model. It has undergone several updates and changes, and some of these cyclical relationships are not very well investigated. Some of them has been confirmed, some of them are, there have been critics on these cyclical relationships. But in principle, this fear avoidance model has gained very much attention. If you look at behavioral studies, there is really good evidence and strong evidence that pain-related fear is an important and a strong predictor of disability in chronic low back pain. There were several studies. Here are a few examples. And there was also a first attempt, 2012, to show this fear of movement phenomenon on the brain level. This was done by a German research group. I will come to that later, but in the end they showed that there are no classical activations in the fear network which are associated with fear of movement. So they concluded that this is maybe another sort of fear, not a classical phobia or something like that. It must be something different, because it does not, or in this study, it did not provoke activations like you see in spider phobia, for example. But I will come to that later in detail. So for the assessment, there are several self-reports, those questionnaires to assess self or fear avoidance. There is the Fear Avoidance Beliefs Questionnaire, very popular, which has two subscales, work-related subscale and an activity-related subscale. Then there is also the Fear Avoidance Pain Scale, Fear of Pain Questionnaire, Pain Anxiety Symptom Scale, and the TAMPA scale, which is also very popular. At the moment, there exist several versions of this TAMPA scale. So the original one was with 17 items. Now there are 13 item versions or 11 item versions. And there is also a special version for the general pain-free population, because it has been shown that fear avoidance beliefs or kinesiophobia can already exist in the pain-free population. And there was a review from Lundberg and colleagues. He criticized that those questionnaires, they all have different fear constructs. So the one is missing here. Oh no, the one here, pain anxiety symptom scale, measures pain anxiety as a psychological construct. Kinesiophobia measures fear of re-injury or kinesiophobia, and this one measures fear avoidance beliefs. So it seems that there are different psychological constructs, and we don't really know how they are related. And they also show very weak construct validity. So this was the main criticism of these authors. So they said that more theoretical research is needed to support this construct validity and thus the measurement of pain-related fear. So again, this was the first study, fMRI study of the German research team, and they showed potentially harmful movements for the back in form of pictures to the participants within an MR scanner. So they showed those pictures. These were selected from the FODA database, which is a FODA database where patients with chronic low back pain rated those stimuli according to the harmfulness of the respective movement. And they selected the top most harmful movements for the back. And they assessed fear of movement with the TAMPA scale and classified the patients into high and low scorers by means of a median split. And yes, as I mentioned, they were in the MR scanner during an fMRI sequence. These pictures were shown in a randomized sequence. And the hypothesis was that, yes, if this fear of movement is a fear or a phobia or anxiety related, then we must see some effects in the fear network of the brain. And as you may know, there are several structures which are related to the fear network in the brain. So there's the amygdala, a key region, a key player of the of threat processing of the fear network. Then also the striatum, the insula, and also prefrontal region, like the orbitofrontal cortex, which also plays a major role in phobias. So they hypothesized that there must be some differences when they contrast the pictures or the conditions between the high scorers and the low scorers. And this was the result. They did not see any, this was a whole brain analysis, they did not see any activation differences between those groups. Only, as you can see here, this was in parietal and temporal fusiform gyrus, but not in fear-related brain regions. So they concluded there was no significant activation in the fear network. So they also concluded, which is that the fear avoidance model must maybe undergo several adaptations only because of these brain results. So I don't agree with this, because brain research can't have such a major role in such interpretations. So there was some critics, also published in Pain 2012, and the main critics, these 2 points were that those pictures, are they really suitable to activate or to induce fear of movement in those patients? And the other critique was that it has been shown that fear avoidance beliefs already exist in the pain-free population, so healthy subjects are not really a good control for this, or at least you have to control for fear of movement in the healthy population. So these were the two main critics, and we started in 2015 to to do it better than not with pictures, with video clips. We used video clips, and we started first of all with a pain-free population. So we had 26 healthy subjects, and the exclusion criteria was no back pain or recurrent pain within the last 6 months, and a history of psychiatric and neurological disorders. And we also took the FODA database, and we took the most 3 harmful activities which were rated by the patients, and these were vacuum cleaning, lifting a flower pot, shoveling soil, for instance. And we also chose as a control walking, which were rated as less harmful in the patients, so walking straight, down the stairs, up the stairs. And we recorded 4 seconds, so the potentially movement was in the middle, about 2 seconds. Each of these video clip was 4 seconds. And we also used the Tampa scale of kinesiophobia, and did it the same way, splitted it in high and low scorers, and this was the special version of the TSK for the general population, which assess fear avoidance or fear of movement in pain-free individuals. And what we saw was quite interesting. So first of all, we observed strong activity in the fear network. This was a whole brain analysis, and we observed activity in the amygdala during the watch. The patients watched the potentially harmful video clips compared to the harmless video clips. And the second interesting issue was this nice positive correlation. Here you see the low scorers and the high scorers. And we had in the left amygdala, we observed a significant positive correlation between the level of the TSKG score as a fear of movement and the signal change in the left amygdala. So this was a first indication that there is a correlate or a neural substrate, must be something related with, somehow related with the fear network. Then we also took another measure or analyzed another measure, which is named functional connectivity. We took the amygdala as a seed region, and then we observed, after the result was that we observed a significant functional connectivity change between the amygdala and the region of the prefrontal cortex, an anterior region of the cingulate cortex, which is called perigenual anterior cingulate cortex. And here we saw or observed an inverse correlation. So the higher the TSG score was, the lower the functional connectivity. So this was the first indication that besides the evidence that fear of movement exists in the pain-free population, so we also add support on the brain level that there must be something different. So this was the conclusion. Fear of movement and related beliefs do exist in the general pain-free population. We could show that on the brain level. And fear of movement is associated with responses in the amygdala and the functional connectivity to an anterior region of the cingulate cortex. So this could be maybe a brain marker of vulnerability, but you also have to interpret these facts with caution, because it's only correlative and it's not causal. So we have first to do maybe longitudinal or apply longitudinal design to show a causal effect or pre and post treatment effect. But it was a first indication. Then we go on and did the same in chronic low back pain patients. And we had 20 patients suffering from unspecific chronic low back pain. That's important. The specificity was ruled out by the clinician. And we did the same experiment. We took care that both groups are age and gender matched, and both of them filled out the Tampa scale of kinesiophobia. The healthy, the G version, and the other one, the normal version. And we did a whole brain voxel regression analysis. So you can do a classical regression analysis with metric variables, you can also do it with voxel. As you may know, we divide the brain in MR or in fMRI into voxels in the 3D space, and dependent on the resolution, you have 100,000s of voxels. And the main aim here was that we feed or put the TSK scores of all participants here into the model, and we were interested in brain activity in voxels, which showed a positive, as an example here, such a positive or a negative correlation between brain activity and TSK score. So this is a very conservative analysis if you take the multiple comparisons into account. So you have to correct for the multiple comparisons, and then it would be a very nice result if we see some correlations within certain voxels with the TSK score. And indeed this was the case. So this was the first analysis where we did not distinguish between patients and pain-free individuals. So all subjects were in this analysis. And first of all, we observed voxels or brain activity in certain regions, which showed this. Here's an example of the left insular cortex, such a nice positive relationship. And these were the only regions in the brain which showed such a positive relationship. And this was mainly, these were fear-related regions: insular cortex, orbitofrontal cortex, also parts of the amygdala or the striatum. And this was the first indication that yes, we can go on with the analysis. And the main hypothesis was, or we aimed to see differences between chronic low back patients and healthy controls. But this was not the case. So we did not observe a significant activation difference between the two groups. But we observed an interaction effect. So this is the interaction effect. This means that if we put both groups into the model and the TSK score into the model, we observed a significant interaction effect in this manner, that the chronic low back patients showed a positive relationship, a strong positive relationship between the left, a certain part of the amygdala, the dorsal part, and the TSK score, and the controls did not show such a significant relationship. And this was the only region in the brain which showed such interaction effect. So this could indicate that this part of the amygdala is somehow related to fear of movement, especially in the chronic low back pain patients. So the neural substrate of fear of movement, of the pathological fear of movement. Then we did another study where we were interested in only the functional or the connectivity between two key regions of the fear network. The first one was the periaqueductal gray, which is a gatekeeper of nociceptive nociception transmission, and is thought to be a key role in pain modulation and in the descending pain system, and the other region was the amygdala. So we were only interested here if this functional connectivity is somehow related to the fear of movement level. So the hypothesis was, because these two key regions are interconnected and play key roles in the PAG in anti-nociception and pro-nociception, and the amygdala in fear, and also the amygdala plays a key role in the modulation or the emotional modulation of pain. So we thought, okay, here must be it's a high probability that we maybe can observe here an effect dependent on the fear of movement level. So here again, you see in yellow the amygdala and the PAG in red. And first of all, we compared the functional connectivity, the strength of the functional connectivity between those two groups. So here is the strength of the functional connectivity between the amygdala and the PAG. Here the potentially harmful activities and the neutral activities. And first of all, we observed a strong high strength of connectivity in the healthy control group, and there was a significant difference to the patient group in the connectivity between these two regions. So it seems that the patients had significantly less functional or weaker functional connectivity between the amygdala and the PAG. And then the most interesting thing was then we went on and investigated the relationship. Here is only the analysis with the patients, and we observed the significant negative correlations between the strength of connectivity and the TSK score. So the higher the TSK score was, the higher the TSK score was, the lower was the connectivity between these two regions. So that could indicate that high connectivity or a strong connectivity between these two regions is healthy, more healthy, and less connectivity can be problematic, and this depends on the strength of fear of movement. So this was the first indication also here that fear of movement modulates key structures of pain modulation and emotional modulation. So these were these 3 fMRI studies in the last 3 years, and they show that we show a correlative relationship. So fear of movement is represented somehow in fear-related brain network, in the fear network brain network of pain-free and chronic low back pain patients. And we also identified a structure in the amygdala, the dorsal amygdala, which seems to play a major role in fear of movement in patients. And also connectivity between key regions of the pain network or fear network, amygdala and PAG. We also observed their effects dependent on the level of fear of movement. So we can conclude also here that brain research can add evidence to behavioral studies, which already show that fear of movement is a strong or is an important predictor of disability in chronic low back pain. And as I said, we have to interpret this with caution, because at the moment these relationships are of correlative nature and not causal. It would be nice to, in the future, also maybe with Dr. Lacher, to see if the treatments can change those brain mechanisms, and this would add additional support or to the success of those treatments. Finally, I would like to talk a little bit about our newest project, because machine learning is now, or deep learning is now, very popular, very sexy, and as you may know, you use machine learning in everyday life. If you open Google Maps or if you drive a modern car. And we can also use this technique in brain research. And what's going on at the moment here is very interesting and fascinating. So the main aim of machine learning in fMRI is that we do not infer, or what we have done with conventional fMRI studies, we do not infer brain activity dependent on a model, like we model an external stimulus, like a painful stimulus, and we would like to see which voxel is then active in the brain. We do it the inverse way. We try to predict the state or the external state dependent or based on brain activity patterns. And we are not longer interested in activity in voxels in certain regions. We are interested now in the pattern of activation. And this makes sense because the brain is not activating some regions at some point, because the brain is interconnected and shows highly interconnected communication, crosstalk. And so it makes sense to analyze the patterns instead of single regions or single voxels. And this is an example how such a classifier works. So we present Homer stimulus B, and with different samples we label them stimulus B, and this provokes certain patterns of activity in the brain. And we present him some examples with stimulus A. And then we apply a certain classifier here. For instance, I can't go in detail, but for instance, a support vector machine. The main principle of this machine is that the support vector tries to find a function which maximizes the distance between those patterns. So he learns continuously, and this is only possible because of the new, or the computer power we have now. So he learns a function to differentiate those patterns that are associated with the different stimuli. And then we can go on, after the stimulus or the classifier is trained, we can go on and present him a completely new pattern, out of sample pattern, and he will tell us, okay, this is stimulus A, this was stimulus A with a certain probability. So this is the basic principle of classification. And imagine now we would not like to predict, so that's a real prediction. He predicts the stimulus. And we would not like to predict stimulus B, we would like to predict a questionnaire score. And as I said in the beginning, we have different questionnaires which show different, or which has, or seem to have different underlying constructs. And now if we go on and only focus on the fear network or brain activity in the fear network, we can see if we can predict the questionnaire score based on fear-related brain activity patterns. And the neural source, or the patterns, are always the same across those different questionnaires. This would indicate that there is a great overlap of these constructs, because if the same brain patterns and the same brain region are predicting the same the level of scores across those questionnaires, this would mean, okay, we can talk about the construct pain-related fear and not about the other constructs like fear, kinesiophobia, or fear of pain. It makes no sense because it's all somehow the same. Because we know they do overlap at a certain stage, and they all are somehow associated with avoidance behavior. And this was the newest study now. So for each question, we had several questionnaires. The patients filled out several questionnaires. And for each of the questionnaire, we computed a prediction model. So here, for instance, you see the FAB-Q, and here you see the true labels, so the true questionnaire labels, questionnaire scores, and here the predicted labels of the classifier. And as you can see here, the classifier was quite good in predicting the level of the questionnaire only based on the fear activity, on the activity patterns within the fear network. And for instance, here you see the weights of the conditions. So the classifier, or the harmful conditions, were more or had more neural information that went into the prediction model than the harmless. So it seems that the harmless condition, it seems that the FABQ is sensitive for those potentially harmful movements. And here you see the region weight, so which regions in the fear network contributed most to the prediction model of the FABQ. And it was quite interesting if you compare this to the other models. Here you have the Tampa scale of kinesiophobia. You first see different sensitivities of the conditions. Here, for instance, there was almost an inverse relationship. This is the TSK11 somatic focus subscale. Here, activity avoidance subscale. And what's quite interesting the main point here is that there were different regions across the different questionnaires which went into the prediction models. So that means that those questionnaire constructs have different neural sources. And that also means that we cannot talk always about pain-related fear, or we can talk about pain-related fear, but we also have to consider that there are maybe differences across those different questionnaires, or the fear constructs in those questionnaires. So for instance, if you see the Tampa scale of kinesiophobia, there was a strong contribution of the orbitofrontal cortex to the prediction model and not the amygdala. And that was quite interesting because we know that the OFC plays a major role in phobias. And the construct the Tampa scale measures kinesiophobia. So this must be somehow related. So we can use machine learning to better understand or to dissect emotional or psychological phenomena where it is not so clear where or how the different constructs are constructed. That's it. This is the team, and thank you very much for the attention. Jetzt muss ich schnell.
Dr. Meier, I think probably everything is clear. Nobody would have a question on that. No, this was research at the highest level, at the highest standards. I think that's important to know. And maybe before I give you the microphone to ask your question, maybe Dr. Meier could have a word or two on the surroundings of his work, on his networks, so that you know how one can come to these data and to this research. It's an amazing thing. So thank you very much.
Thank you. What was the question? Sorry.
Maybe if we could talk a little bit about the bulk risk and about the connections you have, the international network where you are doing this research.
Okay. Yeah, we have, our group is situated or located at the Balgris campus, and as you may know, Zurich has a lot of MR scanners, and we get even more now because the campus itself gets a 7 Tesla scanner and a 3 Tesla scanner, so we have enough facilities and also work power to answer such questions. And we have collaborations, for instance, with the psychiatric hospital, the PUC in Zurich, with Erich Seifritz, maybe you know him. And we have other collaborations with also with the Hochschule Luzern regarding other projects, because this is only the psychosocial parts, but we are also interested in how the spine itself is represented in the brain. So maybe you know the experience of Penfield in the 30s, and he tried to find the body representation on the sensory motor homunculus, and there the representation of the back is not very detailed. So we don't know if, or we would like to see if there are differences in the representation of the back. If you stimulate thoracal parts and if you stimulate lumbar parts, we do not know if how the representation is in the somatosensory cortex. And we would like to, because in the last 10 years there was a hype on fMRI and brain research, and we see differences here, functional connectivity changes here, but we do not really know what this means. And we would like to now to go on and to make a relation to peripheral aspects. So, for instance, we know that there are reorganizations in the brain due to chronic pain, but we don't know why and how, and we would like to see if we can, for instance, relate spinal movement of chronic low back patients to the mapping of the somatosensory cortex, for instance.
Thank you very much. Ars longa vita brevis, as the Latin says. So, if I may give you the microphone.
Thank you. First of all, let me congratulate you to this very interesting research.
Thank you.
May I ask a little question? If I understood you rightly, the second lot of data was elaborated on the same group of patients than the first. If I understood it also rightly, it was 20 in each group, and you had 12 women in the one and 7 in the other. So unless you think women and men are the same, you have a bit of a problem of group homogeneity, don't you? It's a bit of a... how do you correlate for type 2 statistical error?
Sorry, yes, good questions. Actually, it was the same experiment. So it can lead to misunderstandings. It was not a second study population, so we measured both groups at the same time. So, but we did first publish the results on the healthy and then the comparison.
Is there another question, Andre?
You showed this relation between the different activated regions and pointed out that the different connectivity might be the reason for different modes of kinesiophobia. Do you think this is functional connectivity so that it can be changed by learning, or do you think it is anatomically given by birth?
Yes or no?
A good question, because we know that the brain is very plastic, and I think the brain can be changed at every level. Okay, we don't talk now at the brainstem, but I also think at the brainstem level there are plastic changes, and so I think the distinction between functional connectivity and anatomical landmarks is quite also a technical or a philosophical question, because we know that if those gray matter changes, and we also know that these gray matter changes are then also somehow related to functional connectivity changes. And so I can't answer this question definitely, but I think it's changeable, and especially if you look at the chronic low back pain patients, it would also be the reason why we should do more longitudinal research or treatment-based research to see those effects, those changes after treatment, which would indicate that yes, we can change those constraints.
Thank you very much. Maybe a last short question from my side, because I'm a pain practitioner like most of the persons around here. So I would like to ask the question with Menno Sleuter is usually asking. He says, where do we go from here? So that would be my question too. Will we come to a point someday where we can apply this kind of diagnostics in daily practice?
Yes, good question. I think at the moment, we can go this way. So far that at some time we are able to measure brain activity and then to do diagnostics based on brain activity. But I think this is a long way to go because fMRI is, or MR is, it's at the moment it's too costly and it's too big, and so the devices have to be much smaller, or there must be another measurement device like functional NIRS, which is also very promising. But we need, first of all, those devices which are then show the efficiency to do such diagnostics. And the other thing is, at the moment fMRI is also very expensive, and it makes only sense for basic research and not for diagnostics or clinical diagnostics.
So thank you very much again, Dr. Meier.